These are our water potential notes, and this is going to go into the AP Bio Spiral Notebook. So you've already taken notes on diffusion. We know that diffusion is the spreading of particles of a substance along its concentration gradient. So it's going to be moving from a higher concentration to a lower concentration. Diffusion happens naturally, so we don't need energy for it, and every substance is going to move along its own concentration gradient. Passive transport, diffusion across a membrane. We don't need energy. We just have very small molecules or nonpolar molecules that can move in between the nooks and crannies of the molecules of the membrane. Facilitated diffusion, we have diffusion that's helped with transport proteins. So the transport proteins are embedded in a cell membrane, and they basically act as channels to allow a substance to diffuse without energy along its concentration gradient. And then we have active transport. Substances are moved against their concentration gradient. Because they're moving from lower concentration to higher concentration, we have to force them to do that. That's not natural, so it requires energy. Osmosis is the diffusion of water. So why does water want to diffuse? We looked at the fact that it also wants to move from higher concentration of water to lower concentration of water. And solute has a tendency to attract those water molecules. So water potential, oops, wrong button, is once again related to these terms. And so we've seen these before, and we're writing them down again, so chances are they're important. Right? Tonicity is comparison of molarities. So we're comparing the molarity of, say, a cell and the solution that it's in. Molarity is a measure of solute concentration. When we talk about concentrations of solutions, we are comparing the amounts of solvent versus solute in that solution. So you can have a solution that's very concentrated, or you can have a solution that's very dilute. So movement of water, diffusion of water is osmosis, and water potential is looking at that property of a solution that predicts in which the water will flow. The property of a solution that predicts in which direction the water will flow. Okay, so water potential is dependent on how much solute you have and how much pressure you have. I think I worded that a little bit weird. It's dependent on solute, concentration, and pressure. That's probably how I should have worded that. Water potential is dependent on solute, concentration, and pressure. Fix that in your notes. There is an equation that we can solve to find a numerical value of water potential. So the little trident, pitchforky looking symbol, that represents water potential. So water potential equals pressure potential plus solute potential. Water potential equals pressure potential plus solute potential. So now I am on that same page. I'm just underneath that page. I just drew a little line to kind of separate my thoughts. So we have 3 cells here. First cell, second cell, third cell, all of these, think of them kind of like the mystery solutions, the dialysis tubing simulated cells that we did in the science lab. So this would represent a beaker, and the beaker is full of distilled water, and then we put our simulated cell in. Remember that one of our cells ended up being very swollen, and we said that that extra mass came from all of the water entering into the cell from the solution that it was sitting in. That cell would be hypertonic to the solution. The cell is hypertonic to the solution. So this cell is hypertonic to the solution. The cell has a lower water potential. So water potential is a property of a solution that predicts what direction will the water flow. The water is not going to flow inside the cell to outside the cell. So the cell has lower water potential. It does not have, the water that's in the cell does not have the potential to move anywhere. Water from the solution moves into the cell. So the water from outside, the water from the solution it's sitting in, moves into the cell. The cell swells, and we saw that. Our dialysis tubing simulated cell, it got swollen. if it swells enough, it can break. The cell membrane can actually pop, it can burst, and that's called lysis. All right, in this second picture here, pay attention to my arrows. I've got water moving in to the cell, and I have water moving out. This arrow shows water moving in, this arrow shows water moving out. The cell is isotonic to the solution. Isotonic to the solution. So water's moving in and water's moving out. The water still moves in and out, but there is no net change. So remember, we talked about this as being like a dynamic equilibrium. So in here, this relationship is isotonic. Finally, I have this cell over here, and we have water that's moving out of the cell. So this would be like my example cell that I did after you guys did your dialysis tubing cells. The cell is hypotonic to the solution. The cell is hypotonic to the solution. The cell has a higher water potential. Water potential is the potential of the water in a solution to move. And so here, it has lots of potential to move out of the cell. So the cell has a higher water potential. The water leaves the cell and moves into the solution. The cell shrivels, and in living cells that's called plasmolysis. So this cell has a higher water potential. The water has lots of potential to move, and it's moving. It's leaving the cell. Water leaves the cell and moves into the solution. The cell shrivels, and that's called plasmolysis. Solute concentration affects water potential. And if you don't understand my abbreviations, write it out. An increase in solute makes water potential decrease. So the more solute that is in a solution, the less likely it is for that water to leave that solution because it has less water potential. A decrease in solute makes water potential increase. So if there's not a lot of solute in there, then that water has a lot of potential to move. Right, now I am on the right-hand side page. At the top of the right-hand side page, here's some more terms that we need. Turgor pressure, pressure created in a plant cell when a cell membrane pushes against a cell wall. Turgor pressure is pressure created in a plant cell when the cell membrane pushes against the cell wall. Flaccid is a limp cell due to water loss. Flaccid would be a limp cell due to water loss. Plasmolysis, the cell membrane shrinks away from the cell wall due to water loss. Plasmolysis, the cell membrane shrinks away from the cell wall due to water loss. Aquaporin. An aquaporin is a transport protein specific for water. So, facilitated diffusion means that we're helping things to move. We're helping things to diffuse. And aquaporin is an example of a transport protein that allows for facilitated diffusion. So water molecules are small, H2O, so some of them can squeak through that cell membrane on its own. But when I have this big, huge channel, that aquaporin creates a nice big hole, if you will, in the cell membrane, and lots of water can move through. So it helps to facilitate the diffusion of water. All right, now I'm underneath those terms, still on the right-hand side. Pure water in an open container has a pressure potential of 0. Pure water in an open container has a pressure potential of 0. So if I just have a beaker of water sitting out there, its pressure potential is 0. So we can just put a 0 into the equation on that if it's a beaker or a container sitting out in the open. But that's not necessarily always the case. So for example, a plant cell is enclosed and it has a cell wall around it. So here is a beaker of water. Here is my plant cell, and my cell wall is this blue thick line that I have here. So my cell wall is this. Inside I have the cell membrane. So I have a hypertonic solution inside the cell. It is a hypertonic condition. So water moves in, but it can only move in until that swells up and pushes against the cell wall. So this arrow is showing that cell membrane pushing against the cell wall. The cell wall pushes back because it doesn't push against and then pop the cell wall. The cell wall is a structure that helps give the plant its structure. It helps give it support. So as the cell membrane, which is swelling, pushes against, the cell wall pushes back against the cell membrane. So as the cell takes in water, the cell swells and the membrane pushes against the cell wall. That adds a pressure potential inside that cell and helps to keep that cell from bursting. But then once again, that cell wall is going to provide pressure potential. Reverse osmosis uses pressure in order to give you distilled water, if you will. So here's my selectively permeable membrane. With my selectively permeable membrane, adding pressure to it is going to force the water out. So I might have a very concentrated solution over here. Let's say I've got a lot of solutes in here. It has minerals, it has salt, sugar, whatever you want in here, but I want pure water. Remember, the water is attracted to the solute, so it's going to go from where there's pure water to where the solute's at. It's going to move along its concentration gradient from higher water to lower water. Well, if I add pressure, because this membrane is selectively permeable, the only thing that can cross is the water, it ends up forcing the water across the membrane, and then I have water that's moving from lower concentration of water to higher concentration of water. But I need that pressure in order to force that to happen. I need energy. This is kind of like active transport, if you will. We're adding energy to force the water against its concentration gradient. Pressure affects water potential. If you increase the pressure, that's going to make water potential increase. An increase in pressure increases water potential. If you decrease the pressure, it's going to decrease water potential. A decrease in pressure decreases water potential.